Welcome to this virtual training session for the Carl Zeiss LSM 880 with ARIScan Confocal Microscope. My name is Jason Kirk, and I am the Director of the Optical Imaging and Vital Microscopy Core here at the Baylor College of Medicine. Today we are going to be providing a detailed discussion of how to operate this microscope. In this video, we will discuss operating the confocal, from powering it on through collecting and saving a three-dimensional confocal dataset in 3 colors. Before we begin, if you have not already watched the Introduction to Confocal lecture, please find a link here and in the description below. It is a prerequisite to watch this before you take the training. So without further ado, here's a brief overview of the major components of the LSM 880 with ARIScan. From left to right, we have our ARIScan image processing computer. Next is our HXP 120 epifluorescence light source and instrument power buttons. On the isolation table are confocal and ARIScan modules, coupled into our inverted microscope with full incubation chamber. To the right is our motorized stage joystick and remote touchscreen display. And finally, to control the instrument, our host computer running the Zen software. To power on the LSM 880, locate the remote control unit mounted on the electronics rack directly to the left of the instrument. Turn on the main switch. Then turn on the systems PC and component switches. Next, power on the HXP 120 epifluorescence light source. Confocal microscopes use lasers for excitation, so we use a white light source like the HXP 120 along with filters to visualize our fluorescence through the eyepieces of the microscope prior to scanning with the confocal. Power on the host PC, located on the computer cart directly beneath the monitor and keyboard. If you are going to be using the ARIScan, power on the offline workstation as well. When the host PC finishes booting, it will log into Windows automatically, and you'll be brought directly to the desktop. Double-click on the Zen icon to start the confocal software. When the confocal software loads for the first time, you'll be confronted with a login screen that will ask you if you want to start the system or use the software for image processing purposes. We're going to click Start System. Once you click Start System, the instrument will go through an initialization routine where it will check every motorized function and make sure everything is talking correctly. When the login screen disappears, the software will be ready for use. The Zen software is broken out into 3 key areas. The toolbars along the left-hand side of the screen control every motorized function in the microscope. There are 4 tabs across the top of this toolbar: Locate, Acquisition, Processing, and Maintain. In this training, we will be focusing on the Locate and Acquisition tabs. Images will be displayed in the middle section. Along the far right-hand side is a catalog of any images or documents that you might have currently open. To complete the power on routine for the LSM 880, we want to make sure the lasers are powered on and warmed up appropriately. To power on our lasers, we're going to navigate to the Acquisition tab. And from the Setup Manager, we're going to expand the blue laser dialog. Here you'll find a list of every available laser we have on this system, as well as the properties of each individual laser. To power on each laser, toggle the power state from off to on. The argon laser is a unique laser containing lines at 458, 488, and 514 nanometers. This laser has a very specific warm-up routine that must be followed. Once the power state is toggled from off to on, this laser will ignite and then go through a 5-minute mandatory warm-up countdown. The laser is not usable during this time. For time purposes, this process has been sped up. Once the warm-up time is complete, the laser will ramp up to full power. If your intention is to use the images from this confocal microscope for quantitative analysis, you must let these lasers warm up for a minimum of 1 hour before collecting any data. This is to allow the laser's proper warm-up time to stabilize. Toggle on the remaining lasers you require for your experiment. You'll notice the 405 nanometer laser does not have a power on toggle button. This is because this laser is direct modulated, and it can be activated later on when we select it in our imaging setup. As an alternative to powering the lasers on manually, the OiVM core provides pre-configured experiments. You can access them from the Experiment Manager at the top of the Acquisition tab. Simply click on the Open Folder icon to view a list of the available configurations. Each experiment is a stored group of settings that saves the Beampath configuration and acquisition settings for each channel. Experiments are named based on imaging mode for ARIScan, ARIScan Fast, and traditional confocal, as well as the laser line combination used to excite the fluorescence of each channel. Please note that each of these experiments is designed for sequential imaging only. If you require simultaneous imaging or any other imaging modality, please see core staff and we'd be happy to help configure the microscope with you. To load the parameters for a particular experiment, simply click on the required configuration, and all the parameters that are stored with that configuration will be applied to the software. This includes the power state of the lasers, the configuration of the filtering system, as well as a variety of baseline settings for adjustments we will be discussing later on in the training. This is a great little shortcut for powering on all the lasers you require and setting up the configuration on the confocal in one single step. To mount our samples on the microscope, first we'll need to open the incubator. Open the two bay doors on the front of the incubator. Next, slide the top doors open. With one hand bracing the eyepiece column, gently push back on the transmitted arm. With the incubator open and accessible, we can now prepare our stage for sample mounting. Ensure that there is no sample holder mounted on the stage and that the area around the objectives is clear. We need to decide which objective lens we will use for imaging. This choice depends on the resolution and field of view you require for your images. This microscope is outfitted with 6 objectives. The first 3 positions are low magnification, dry objectives at 5, 10, and 20x. The 5 and 10x are typically used for screening samples, and occasionally for imaging larger specimens. The last 3 positions are high magnification, high resolution objectives for aqueous specimens using water immersion, and for specimens mounted in high refractive index mounting mediums, like prolong or slow fade, using oil immersion. There are a few key pieces of information you need to know to appropriately select an objective lens. The first is what is the size of the structures you are trying to resolve. Are you looking at cells interacting with other cells, or are you interested in the dynamics of subcellular organelles? The answer to this question will determine how high of an NA, or numerical aperture, you require. The NA directly influences the resolution of the lens, so choose an objective that allows a high enough X, Y, and Z resolution for your experimental needs. Field of view is also a concern, and will depend on how much area you need to cover in a single image. Resolution is more important than field of view, so if your resolution needs demand a higher magnification objective, we can increase the field of view later on by using the tiling feature in Zen. Please familiarize yourself with the resolution capabilities listed for each objective. For this training, we are going to use the dry 20x 0.8 NA objective to keep things simple. In another tutorial, we will discuss using immersion objectives. To select the correct objective, locate the TFT display to the right of the microscope and make sure that you are selected on the home screen. On the left-hand side of the display, click on the Microscope tab. From the Control tab, select Objectives from the top menu. Then select 20x from the objective list. This action will move the objective focus down away from the specimen plane and then rotate the turret to the objective you chose. If an immersion optic was in place, the TFT will display a notification warning you that you are about to move from an immersion lens to a dry lens, or vice versa. This mechanism protects the objective from coming into contact with immersion fluid if they are not designed for it. As a general rule of thumb, we do not recommend changing objectives between immersion and dry without first removing the sample. We will discuss this in greater detail during the immersion optics tutorial. With no specimen in place, it is safe to click Done. If the objective moves its focus back up toward the specimen plane, we will need to adjust it. From the right-hand side of the TFT, click Load Position to move the objective to the lower limit of the focus drive. Then click Set Work Position. This is the safest place for the objective while we mount our sample. In addition, please remember to move the objective to the load position anytime you change samples. To mount our specimen, we will need a sample holder that can be inserted into the universal slot on the motorized stage. The LSM 880 has 3 choices available for specimen holders. The first is a holder for multi-well plates. The LSM 880 is capable of imaging with multi-well plates, but only if they have 170 micron cover glass bottoms. No plastic. The second is best suited for 75 by 25 mm slides, as this holder has restraint clips to hold the slide into place. This holder is useful for multi-position or tiling experiments when using immersion objectives, as the clips prevent any potential movement. The third option is a universal holder that accepts slides as well as 35 mm dishes, chambered cover glass, or custom sample holders. For this training, we will use the universal holder. Insert the sample holder into the machined well by aligning the red dot painted on the stage with the red dot on the holder. In this lower left corner, you will notice 2 leaf springs mounted into the recessed portion of the stage. These springs put tension on the holder to help secure it snugly into place. To mount this holder correctly, you will need to insert it into the stage with the red corner first, applying pressure in a downward motion to compress the springs with the edge of the holder. If the holder is in place correctly, you will see the 2 leaf springs in the lower left corner compressed against the holder. Here is an example of an incorrect installation of the sample holder. You can see that there is a considerable amount of wobble when the holder is installed this way. This will make it impossible to keep your focus or stage position stable. When the sample holder is mounted correctly, use the joystick for the motorized stage to roughly position the sample holder so the objective is centered. Mount the slide into the sample holder with the cover slip down, facing the objective. Keep in mind that on any microscope, cover slips should always be facing the objective. Carefully push the holder jaws together so the sample is held snugly. Use the joystick for the motorized stage to roughly position your section over the objective. You'll notice on this slide that we've used a Sharpie to draw a circle around our section on the back surface of the slide. This will help us easily locate the section later by eye. Now we can move the transmitted arm back down and close up the incubation chamber. I'm going to pause here and ask that you be very careful lowering this arm. There is a laser safety switch tucked into the base of this transmitted arm. If the arm comes down too fast and too rough, this sensor can be damaged. If this sensor is broken, it will effectively disable the entire confocal, so please be very careful when lowering this arm. If you are using the AriScan, we recommend that you close the chamber, as small air currents and temperature fluctuations can have an effect on the image acquisition. If you are using fixed preparations on the traditional confocal, however, feel free to leave the incubation chamber open. To begin visualizing our sample in the eyepieces of the microscope, navigate to the Locate tab in the Zen software. Clicking the Locate tab will put the microscope into a laser-safe state, allowing you to direct fluorescence light excited by the HXP120 to the eyepieces. The Locate tab houses functions that control the epifluorescence and transmitted light paths of the microscope. Under Ocular and Microscope Control, you will find a schematic of the light path in the microscope. Here you can manually change filters, turn on and off light sources, and control their intensity. This inverted microscope is configured for visualizing either a red, green, or blue fluorophore via the eyepieces. As a side note, these filters are used for visualization only and will not affect the confocal settings. Adjusting each of these parameters by hand each time you use the microscope is a bit much to ask, so the OiVM core has pre-programmed shortcuts for you that correspond to the different red, green, and blue filter sets available in this microscope. In this example, we are going to focus on our sample using the green fluorescence. So we just click green from our shortcuts list. When we do this, you'll see the schematic change to reflect the settings programmed by the shortcut. You'll see that the green filter set has been set into place and the light source has been turned on. Moving our attention to the microscope stage, you'll now see the blue excitation that is used to excite our green fluorophore striking the specimen. Check by eye that your sample is positioned correctly on the stage, and update the position using the joystick if necessary. On many sections, you can easily see by eye when observing the objective when the section goes in or out of the illumination cone. You should see the brightness of the illumination cone increase when the section is over the objective, and decrease when it is positioned away from the objective. From here, we can begin to adjust our focus. Familiarize yourself with the focus knobs located directly to the left and right of the microscope body. Each of these knobs has a coarse focus located on the outside and a fine focus located on the inside. Turning these knobs towards the microscope will move the objective up closer to the specimen. and rotating the knobs back towards you will move the objective away from the specimen. You'll recall that we positioned the objective into the load position prior to mounting our sample, so the objective should currently be at the lower limit of the focus drive. While looking through the eyepieces of the microscope, begin slowly rotating the coarse focus knob towards the microscope. To start, you should see nothing in the eyepieces but a black field. Don't worry, this is perfectly normal at this point. As you get closer to the specimen, you should see the entire field of view starting to get gradually brighter and brighter. When you start to detect this brightness change, slow down your coarse focus movements. As soon as you start to see the outline of the specimen, switch to the fine focus knob and continue focusing until the image is sharp. When you have successfully focused your specimen via the eyepieces, use the joystick for the motorized stage to locate an interesting area of the specimen. Ideally, you want to center any area of interest, as the confocal scanner will only see about 60% of the overall field of view you see through the eyepieces. When you find a suitable location, move back to the Zen software and turn off the epifluorescence light source by clicking on the All Closed shortcut. Try your best to quickly position your sample, limiting the amount of time you're exposing the specimen to the illumination light to prevent premature photobleaching. Once our light source is off, we can move back to the Acquisition tab in Zen. After our sample has been mounted and we've located a region of interest, we can begin configuring the software to detect our fluorophores. To do this, we need to create light path configurations for each of our fluorophores. Before we begin this process, we must first have a good understanding of the excitation and emission profiles for the fluorophores we are using. Please familiarize yourself with this information, which can be found in the technical documentation from the manufacturer of the fluorophore you are using. Specifically, you want to know the nominal excitation and emission range of your fluorophores. We must also decide how we want to collect each of these channels. You can collect all the fluorescence from your sample simultaneously, or you can choose to collect each fluorophore one at a time, referred to as sequential acquisition. You'll remember from our Introduction to Confocal lecture that fluorescence emission is Gaussian in its spectral distribution, which means that given enough laser energy and detector sensitivity, you can see signal from many fluorophores just about anywhere in the visible spectrum. Remember that this can lead to cross-excitation and/or cross-emission problems, which is the visualization of fluorescence in areas outside its nominal range. The combination of fluorophores you use will determine the degree of spectral overlap you may encounter. Unless you require high-speed imaging of live specimens, or you have extremely well-separated emission profiles, the sequential method is generally the preferred approach to imaging fixed or immobile specimens. To set up a sequential acquisition in the Zen software, expand the blue imaging setup dialog. At the top of this dialog, you will find a list of tracks. For sequential imaging, a track is defined as a group of settings specific to an individual fluorophore. When a track is selected, the schematic of the light path reflects the filter settings that correspond to a particular fluorophore. In this example, we will be programming a 3-color sequential configuration for RFP, GFP, and DAPI, or a red, green, and blue fluorophore combination. We always want to begin with the longest wavelength fluorophore first. This will avoid any potential cross-excitation of lower wavelength fluorophores in the sample before they can be imaged. Before we begin, let's recall that a confocal microscope is a fluorescence microscope, so the filtering mechanisms found here are foundationally identical to a simple epifluorescence microscope. To detect fluorescence in a traditional wide-field microscope, we need to position a series of filters between the light source and the objective. These filters are often housed in a simple cube, configured to direct the excitation to the specimen and collect emission back to the eyepieces. In this example, we will be looking at a green fluorophore, where our excitation is in the blue range, from 470 to 490 nanometers, and our emission is in the green range, from 510 to 550 nanometers. When we put this filter into place and open the shutter on the white light source, the entirety of the visible spectrum from the HXP120 strikes the filter cube. Here it encounters an excitation filter. This filter is specially coded to only transmit a bandpass of light, a range centered around the fluorophore's nominal excitation. All other wavelengths above and below this range are rejected. In this example, the excitation filter passes between 470 and 490 nanometers. Once this excitation range passes into the filter cube, it strikes a dichroic mirror. A dichroic is a piece of plate glass, angled at 45 degrees, that has special coatings on it that reflect wavelengths of light shorter than a set cutoff wavelength, and transmits wavelengths longer than this cutoff. A cutoff is typically positioned somewhere in between the nominal excitation and emission bands of the fluorophore. In this example, the dichroic is cutting at 500 nanometers, so everything shorter than 500 nanometers reflects at a 90-degree angle and goes into the objective, and everything longer than 500 nanometers transmits directly through. After this dichroic, our excitation light is focused by the objective onto our specimen. Our green fluorophore goes from a ground state to an excited state, relaxes for a brief period, and then emits a photon of light in its nominal emission range, which is 510 to 550 nanometers in this example. This longer wavelength emission light is collected by the objective and makes its way back to the dichroic. Now, because the emission range is longer than 500 nanometers, it will transmit directly through. Once the emission light transmits through the dichroic, it will encounter an emission filter, which, much like the excitation filter, is a bandpass filter that only allows a certain range of wavelengths to pass. In this example, the emission filter transmits between 510 and 550 nanometers and blocks everything outside this range. The emission light is then directed to the eyepieces of the microscope. The imaging setup Beampath is essentially a fluorescence filter set. However, it is a bit more complex. But it still relies on the same three components of a filter set: excitation, dichroic, and emission. Let's walk through the setup of our first track for RFP, or a red range fluorophore. First, select the excitation source. Here you'll find two light paths, one for the visible light and one for invisible light. The visible path directs our lasers from 458 nanometers to 633 nanometers, while our invisible path directs light from our 405 nanometer laser. This schematic is a little misleading, as these excitation paths are completely separate and do not interfere with one another. From the visible light path, click on the lasers icon and check the box next to the 561 laser. This will activate this line for excitation in this track. Then set the laser power to 2%. Because these lasers are monochromatic, there is no need for a separate excitation filter. We will discuss how and when to modify the laser power later on in the training. Next, follow the line from the laser icon to the MBS filter icon. This is our main dichroic, or main beam splitter, MBS for short. You'll hear the term beam splitter or beam combiner used interchangeably with dichroic on many confocal microscopes. Click on this filter to expand the options. Ideally, we want to select a filter that is going to match all the laser lines we will need for all the tracks on the visible path. Since we are going to be collecting a red, green, and blue fluorophore, we need to select a beam splitter that matches the 488 and 561 lasers we will need for our two visible fluorophores. Choose the 488-561 option. While these filters are dichroics, they are quite different from a traditional dichroic. Traditional dichroics typically have a single cutoff value, meaning they can either reflect light or transmit light around a single wavelength. The beam splitters here are multi-line filters, meaning they have a reflection notch coded for each line noted on the filter. A notch is a narrow reflection band where the beam splitter only reflects light plus or minus about 5 nanometers around the lines displayed for that filter. So for the MBS 488-561, this filter will only reflect two bands of light, one between approximately 483 and 493 nanometers, and the other between approximately 556 and 566 nanometers. These are perfect for our 488 and 561 lasers. In addition, emission light between 493 and 556, as well as emission above 566 nanometers, will transmit to the detection system. This is important because that is the ideal range of the emission spectrum for our fluorophores. Since we will design a third track for DAPI, we want to set the MBS for the invisible path to MBS 405 at this point as well. Even though this won't affect the visible path we are currently designing, we don't want these MBS filters changing between our tracks. This will help save acquisition time down the road. At this point, you have successfully directed excitation light to the specimen. Now we want to set up the light path to collect the emission back from the objective. As our emission light is collected by the objective, it transmits up through the main beam splitters. Because this emission signal is between the reflection bands of the dichroic, it can transmit directly through. The next filter the emission light encounters is another beam splitter, which either sends light to the confocal, to the left, or to the ARIScan detector, to the right. Because we are designing a 3-color configuration, we will only send light to the confocal in this example, so we are going to ignore the ARIScan detector for this part of the training. Set this filter to mirror to direct all the light to the confocal. Now on a traditional epifluorescence microscope, our next step would be just to set the emission filter and be done. The confocal pathway is a bit more complex, however. There are 2 choices we need to make here. The first is which of the 3 detectors we will send our emission signal to. And the second is the emission bandwidth, or the range of wavelengths we want to collect for our red fluorophore, and more importantly, what we want to reject. Before we can make these choices, we need to understand how the detection path on the LSM 880 works. Rather than a simple emission filter and detector, this microscope employs a complex spectral array that can simultaneously collect an image every 8.5 nanometers over the entire visible spectrum, a possible 34 channels at one time. The following is an overview of the spectral array on the LSM 880. Once our emission light passes into the detection unit, the light strikes a diffraction grating, which will disperse light like a prism, sending long wavelengths to one side of the array and short wavelengths to the other. The detection pathway consists of 3 physical detectors. The one in the middle is a 32-channel gallium arsenide array with 8.5 nanometer windows covering the entire visible spectrum. This array detector is flanked by 2 multi-alkali side lobe detectors. Channel 1 is for blue shifted fluorophores, and channel 2 is a special red sensitive PMT for near infrared fluorophores. By default, all the light is transmitted to the gallium arsenide array, referred to here as channel S1. And we can electronically combine various points on the array that align with the spectral emission range of our fluorophore. This allows us to manually define the precise spectral range collected. The drawback to this detector is that it has only a single amplifier. So when you adjust the sensitivity, it will be the same for all the channels. If you are using a fluorophore that is very bright alongside one that is very dim, it will not be ideal to use the same detector for both. Therefore, the 2 side lobe detectors, each with their own amplifier, can pick up the slack for the array detector. Light is split between the central array and the side lobe detectors by wedge prisms, located on either side of the emission path, that can be adjusted in and out of the light path to control which wavelength ranges strike which detectors. For channel 1, the edge of the outer slit controls the lower end of the range, while the tip of the prism controls the higher end of the emission range collected. The opposite is true for channel 2, where the prism controls the lower end and the slit controls the higher end of the emission range. There are really 2 main takeaway messages from this discussion. The first takeaway is the order of the channels. Channel 1 collects the shortest wavelength fluorophore in your series. Channel 2 collects the longest wavelength fluorophore. And channel S1 collects everything in the middle. The second takeaway is the choice of gallium arsenide or traditional PMT. Traditional PMTs are not terribly sensitive. They typically have between 15 to 20% quantum efficiency. A gallium arsenide detector's quantum efficiency is closer to 50%, so if you have a particular fluorophore that is weak and would benefit from being detected by the more sensitive GASP array, design your light path so that fluorophore uses channel S1. Back in our imaging setup, since we are currently programming our red fluorophore, we want to choose channel 2 for our detector and turn off all the others. Next, we want to set the detection range for our emission. You can manually enter the values by clicking on the emission range and entering the start and end values for the range you require. Here we will be collecting between 575 and 625 nanometers for our red fluorophore. Lastly, we can choose our pseudocolor from the Color tab, and our track design is complete for our first channel. Before moving on to the next fluorophore, navigate to the Channels tab in Acquisition Parameter and rename this track something more useful than Track 1. Double-click on the track name at the top of the dialog and enter the excitation wavelength we used, 561, then hit Enter on the keyboard. Back in our imaging setup, we want to add another track. This time for GFP, or our green fluorophore, and start configuring. To the right of our track list, click the plus button to add a new track, and then repeat the same process that we used for our red fluorophore, but this time selecting the particulars for the green fluorescence. Before moving to the Beampath, confirm that the setting Switch Track Every is set to line. This will mean that during the acquisition, the instrument will scan each X line 3 times, one for each track, before moving on to the next Y position in the scan. The line mode is the fastest way to switch between tracks during acquisition, but it does not permit any mechanical changes in the detection pathway between tracks. The only changes permitted are which laser line is active and which detection channel is selected. From our laser icon, activate the 488 laser and set its power to 2%. We don't have to change anything with the dichroics, since we set these filters to the ideal settings while programming our first track. You'll recall that these dichroics will pass the emission ranges for all our fluorophores with this combination. Next, select channel S1 as our detector and set the emission range between 500 and 550 nanometers. You can type these values in by clicking on Range next to channel S1. Recall that channel S1 was ideal for our middle fluorophore. Select a pseudocolor and navigate back to the Channels dialog to rename this track to 488. It's back to our imaging setup to program our last track, which is for DAPI. Click plus to add a new track. This time select the 405 nanometer laser from the invisible laser icon and set the power to 2%. Activate channel 1 as our detector. Remember to make sure to turn off all the others. Set our emission range from 420 to 480 nanometers, and then choose a pseudocolor for this channel. Finally, rename the track 405 back in our Channels dialog. Congratulations, you did it! This is arguably the most complex part of using this instrument, so when you're comfortable with this, then you will have come a long way. We now have a 3-channel sequential acquisition setup that is ready for imaging. As we stated earlier, an alternative to programming each of these channels in manually is utilizing the pre-configured experiments the OiVM core provides, that store the Beampath configurations as well as all the other acquisition settings. These are a great baseline for you to program your own configurations, or to use as provided. We do ask that you do not save your own configurations directly to the experiment manager. All these acquisition settings are stored with the image files as well, so if you need to recall a configuration that you have modified, you can reuse it from the image directly rather than using the experiment manager. This way, hopefully no one overwrites our configurations. When we finish configuring the light path for our experiment, either by loading a pre-existing configuration or designing a new light path via the imaging setup, we can begin to optimize the settings for each individual fluorophore. From Acquisition Parameter, expand the blue dialog labeled Channels. Here you will find a list of the program tracks, sorted from long wavelength to short wavelength as designed in our imaging setup. You'll notice that the settings are different for each individual track. This track list allows you to set the order of acquisition, the name of each track, as well as the pseudocolor applied to the image. I highly encourage you to experiment with different pseudocolor combinations. The traditional red, green, and blue tend to be poorly contrasted and particularly terrible for viewers who are colorblind. You'll notice that there is a checkbox to the left of the track name. This indicates that the track will be scanned. Deselect this checkbox to disable a particular track. In addition to the checkbox, you'll also see that the track upon which you are selected is highlighted. This means that the settings that you see directly below the track list correspond to the track highlighted. Make sure that when you are optimizing each track, that both the checkbox is active and the track is highlighted. Each track in the list will have its own independent adjustments for 3 key settings. These are laser power, pinhole diameter, and detector sensitivity. These 3 settings play off each other. For example, making an adjustment to the laser power will influence the detector sensitivity settings. So you will have to balance these in an optimal way. Fortunately, we can fix 2 out of the 3 parameters to start, and depending on the response of the fluorophores in the specimen, we may or may not have to make additional adjustments to them. Going from top to bottom in this dialog, the first adjustment parameter we can fix is the laser power. There is a checkbox to activate the line selected in our imaging setup, and a power adjustment slider to control the intensity of that line from 0 to 100%. The overall power of the laser will directly affect the image quality, but more importantly, it will have its biggest impact on the health of your fluorophores and your sample. Increasing laser power will yield more signal, but it will also induce negative photo effects such as photobleaching and phototoxicity that can permanently harm the sample. Lasers also generate considerable heat when used at higher powers, and that may have unforeseen effects on specimen health. Your fluorophores can only take so much exposure, so we need to be careful about how much laser power we use. For most lasers on this microscope, we recommend starting laser power settings between 1 and 5%. It is recommended to start as low as you can possibly go and still get a reasonable image. If necessary, laser power can be increased as needed to help balance image quality. In this example, we'll start this intensity parameter at 2%. The OiVM core has roughly calibrated this against the output of the epifluorescence pathway. So if you can see your fluorescence easily via the eyepieces, then 2% laser power should be enough to start. The second parameter is the diameter of the confocal aperture, or pinhole. This value determines the optical section thickness, or Z resolution, detected by the confocal. The software reports the theoretical section thickness, as well as how many area orders the aperture passes. To set this parameter, make sure you are selected on the longest wavelength track programmed into your configuration. The LSM 880 has only one pinhole diaphragm for all tracks, and as resolution is a function of wavelength, setting this diameter to the longest wavelength track will not negatively affect the signal of the lower wavelength tracks. Click on the 1AU button to automatically set the preferred diameter. You'll recall from our Introduction to Confocal lecture that setting the pinhole diameter to 1 area unit allows only the signal from the central area order to transmit to the detector. The bulk of your in-focus fluorescence signal comes from that central area order, and we can reject out-of-focus light from higher area orders without tremendous loss of signal. The 1AU setting is a compromise value, however, between reducing the detection of out-of-focus light and maximizing signal to our detector. This graph illustrates the effect of the pinhole diameter on the signal intensity and resolution. Signal is the green line in this illustration. If we start with our pinhole wide open, we have a state where we are collecting the maximum amount of signal, but have the worst possible Z resolution. As we close down this pinhole, you can see that the Z resolution improves greatly, while the signal level is reducing at a much lower rate, a good compromise. All this changes, however, when the diameter reaches 1 area unit. When we cross this threshold, our Z resolution continues to improve at roughly the same rate, but now we start to lose signal exponentially. So again, 1 area unit is a good starting compromise between resolution and signal. Now if you have a stable and very bright signal, you can take the pinhole diameter below 1 area unit and continue to improve your Z resolution. In fact, this will even start to improve your lateral resolution. Alternatively, if your signal is weak and you don't want to increase the laser power too much, you can open up the diameter above 1 area unit to improve the signal level. This is especially useful when you can sacrifice some axial resolution. With those 2 parameters fixed, we can focus our attention on the third adjustment, which is detector sensitivity. The sensitivity of our detector is adjusted with 3 sliders: master gain, digital offset, and digital gain. I want you to ignore the digital gain slider and leave it set to 1. This is an additional electronic gain factor that is only preferred when the analog gain, or master gain, is exhausted. Master gain is the analog voltage applied to our photomultiplier tube, directly regulating the amplification of our signal. Digital offset is the offset value relative to a dark current calibration done for the instrument by Zeiss Service. This value controls the ambient background signal of the detection system. This slider should not be used to correct any potential background signal from non-specifically labeled areas of tissue or autofluorescence. That is real signal, want it or not, and it is important to not eliminate it at this stage. To simplify, the master gain controls where the brightest parts of our signal fall within the dynamic range of the image, and the offset controls the ambient background signal from our detection system. Let's begin optimizing the settings by scanning a new image. First, start by highlighting the longest wavelength track and ensure that the checkbox is active next to the track name. Next, turn off all the other tracks except the one you'll be working on. This is to avoid unnecessary scanning of tracks you're not actively adjusting. Navigate up to our scan buttons located below the Experiment Manager at the top of the Acquisition dialog. Here you will find two buttons that will initiate scanning. The first is Live, which when clicked will begin scanning with a 512 by 512 frame size at the maximum speed for the current field of view. The Live function is useful for quickly updating the scan field so that we can see the results of any changes we make to the detection settings. The Snap button will collect a single image using the programmed frame size, speed, and any additional image optimization steps that the Live scan ignores for speed purposes. Snap can be used to collect a final image. Click Live to begin capturing images. A new image will populate into the display and start updating. From our Channels dialog, turn up the master gain until you see some signal. When there is signal displayed that corresponds to the structures you saw in the eyepieces, stop the live scan. This is now our baseline image. Our next step is to adjust the detection system so that the entirety of our signal falls within the dynamic range of the detector. The dynamic range is defined as the ratio between the brightest and darkest signals the detector is able to see. We ideally want to adjust our detection so that the darkest signal we have is just above the lower limit, and our brightest signal is just below the upper limit. We first need a way to identify those areas in the image that are at the upper limit, or overexposed, and the areas in the image that are at the lower limit, or underexposed. To do this, we need to apply a LUT, or lookup table, to the image that will identify those regions. In the Zen software, this LUT is referred to as the range indicator. You can activate this lookup table in one of two places. The first is in the Image Display toolbar at the bottom of the image. In the Dimensions tab, check the box labeled Range Indicator. The range indicator will pseudocolor areas of the image red where there is overexposure, and color areas blue where there is underexposure. This checkbox is a soft setting, which means the next time you start a scan, the colors will go back to the LUT you set when you loaded the configuration, which is red in this example. To apply a more permanent setting, which is to say one that will stay on until you turn it off, navigate back to the Channels dialog, expand the display arrow at the bottom of the dialog, and check Range Indicator. This setting will make it easier to use the range indicator during the entire process of optimization. Now we are ready to make our adjustments on this channel. The two values we will be adjusting here are the master gain and digital offset. Master gain controls the brightest parts of the image, colored in red, and the offset controls the detector background, colored in blue. Click Live to start a new scan. Adjust your focus to locate the brightest image plane. Begin by optimizing the offset. This value is adjusted in the absence of fluorescence signal. So while we are scanning, we need to first deactivate the laser. If we see a large area of blue pixels, increase the offset value until these disappear. It's okay to have a few randomly appearing blue pixels, we just want to avoid large patches of them. Once an offset value has been found, you can leave this setting the same for the remainder of the imaging session. Toggle the laser back on by checking the box above the laser line. If you see red pixels in the image, decrease the master gain until they disappear. If you do not see red pixels, increase the master gain until just before the red pixels appear. Stop the live scan. An optimal image will have no red and no blue pixels in the final scan. Keep in mind that we are making these adjustments to make sure that our signal falls within the range of detection for this PMT. You are collecting raw data at this point, so do not make adjustments for contrast or the prettiness of the image at this stage. Adjustments like this can be done later, after our final data is collected. Now we can repeat this process on the remaining channels. Activate our 488 track, then deactivate our 561 track. Remember to highlight the 488 track before scanning. Click Live to start a new scan. From the Channels dialog, deactivate the 488 laser and adjust the offset until you have no blue pixels. Toggle the 488 laser back on and adjust the master gain until you are just below the display of any red pixels in your brightest areas. Remember to adjust your focus to make sure you are optimizing to the brightest image plane. Stop the live scan and activate your 405 track. Click Live to start a new scan. Deactivate the 405 laser and adjust the offset until you have no blue pixels. You'll notice that each of these detectors has slightly different offset values. Toggle the 405 laser back on and adjust the master gain until you are just below the display of any red pixels in your brightest areas. Be sure to fine-tune your focus so you are optimizing to the brightest image plane. Stop the live scan. With each of our channels properly balanced, turn off the range indicator from the channels dialog. Then turn on each track in our track list and click Snap to collect a final image. This configuration will now scan each X line 3 times, one for each channel, while moving the Y scanner down the image. Now that we have our channels properly balanced, we need to make some final adjustments to the scan to optimize our image resolution. Expand the Acquisition Mode dialog located just above Channels in Acquisition Parameter. Here you will find the settings for optimizing resolution and field of view in our final scan. At the top of this dialog, you will find the objective currently programmed into the software. Confirm that the objective you are using on the microscope matches what is displayed in the software. This is important because all the calculations the software will perform to measure image resolution will be dependent on the accuracy of this entry. Directly below objective, you will find the frame size of our image. This is how many pixels in both X and Y will be scanned to build the final image. The Zen software uses 512 by 512 pixels as the default frame size. However, if you load any of the OiVM core's configurations from the Experiment Manager, the default frame size will be 1024 by 1024 pixels. So how do we know how many pixels we need to use? To figure this out, we need to understand how our lateral resolution is calculated in the microscope. Lateral, or XY resolution, can be calculated using the Rayleigh criterion, which is 0.61 times the emission wavelength lambda divided by the numerical aperture of the objective. Using this formula, if we assume lambda to be the middle of our detection range, which in this example would be 530 nanometers, the best resolution this objective can achieve is 404 nanometers. This is the smallest object that this objective can resolve. Now all we need to do is set the frame size so that each pixel is 404 nanometers, right? Not quite. We ideally need to sample this objective digitally using the Nyquist criterion, which states that for the optical resolution defined by the objective, we need to sample this 2.5 to 3 times digitally in order to properly represent a diffraction-limited structure. Okay, so what does that mean? That means if our objective can resolve down to 404 nanometers, we need each pixel to be between 135 and 162 nanometers in order to generate an image that adequately resolves down to our objective's limit. Well, nobody wants to have to do math while sitting in front of the microscope, so Zeiss has made this calculation easy for us. Just below the frame size, you will see a button called Optimal, which will automatically set the frame size to the correct Nyquist sampling rate. Click on Optimal to set the proper sampling frequency. You'll see the frame size jump from 1024 by 1024 to 3440 by 3440. This is a major increase in image size. You should look at this setting as the ideal frame size. Any higher than this would be oversampling, and any lower than this and you are not achieving the resolution this lens is capable of. Let's take a look at a real-world comparison of images taken with the default frame size versus the optimal frame size. First, we are going to scan an image at Zeiss's default 512 by 512. Click New to generate a new blank image document so we don't overwrite the image already taken at 1024 by 1024. Next, change the frame size to 512 by 512 using the XY shortcut list in the Acquisition Mode dialog. Snap an image. Now let's generate an image at the optimal sampling frequency. Click on the Optimal button under the frame size setting. For speed purposes, we are going to increase our scan speed to max. More on that later. Click New to generate a new image document. Click Snap to collect the image at the optimal setting. You'll also notice the amount of time that it takes to collect a properly sampled image compared to an undersampled image. Get used to that. Point scanning confocals are slow. Slow. So. Very. Very. Slow. When the acquisition is complete, let's display these side by side. To do this in Zen, right-click on the area next to the Image Name tabs at the top of the image window. Select 3 container from this list, so we have 3 side-by-side slots for images. Click and drag each image into its own frame, with the optimal on the far right, the 1024 by 1024 image in the middle, and the 512 by 512 on the left. Zoom them up to 100% so each pixel matches the pixel size on the monitor. Already you get a feel for how much more data is in the properly sampled image. Now increase the digital magnification so all the images are the same size on screen. There is quite a big difference in quality between the images collected at the optimal settings versus the default settings. Again, the choice depends on what you are trying to resolve. If the structures in the 1024 image are suitably resolved for your quantitative purposes, use that setting. If you are unsure, mainly because you haven't collected any data to attempt a quantification yet, collect some test images at the optimal settings and start attempting to quantify these images using processing software. You want to try to work out your ideal parameters before collecting tons of data. There is nothing more frustrating than getting to your quantification stages only to realize that your data is inadequate. Another feature used to control our final image quality is the speed at which we scan the image. The longer you spend at any one pixel, the more signal is integrated and thus the lower the noise for that pixel. This comes at a price, however. The longer you spend sampling at any single location in the image, the more potential photo bleaching and photo damage you may encounter. So the faster we make the scan speed, the noisier the signal. But when we slow it down, we get an increased probability of photo bleaching. A bit of a Sophie's choice. There is a happy medium, however, that we wholeheartedly support: averaging. To handle noisy images, we prefer to set the scan speed to max and increase the amount of averaging to improve the signal-to-noise ratio in the image. Averaging works by scanning the same X line multiple times and then averaging the result of those scans for each pixel. Averaging is useful because the scan speed is high, so less photo bleaching, and the scanner returns to the same area at different time points, giving the fluorophore a chance to relax between scans. Most fluorophores tend to respond much more favorably to this tactic rather than simply reducing the scan speed. Let's take a look at some images to see the effect of averaging. First, let's select just our green channel and make some changes to our settings to simulate a weak fluorophore. Here you'll see that the detector gain is currently set to 587 volts. Anything under 600 volts at 2% power is quite good signal and rarely requires averaging. So we are going to significantly reduce the laser power from 2% to 0.05% and observe the results. If we scan this channel now, we'll have to significantly increase the gain to compensate, to 812 volts. Let's snap an image with no averaging applied to the scan. Now let's go back to the acquisition mode and turn on our averaging. From the averaging section in the middle of the dialog, click on the number drop-down to expand the options. Here you'll find values for 1, which is no averaging, 2, 4, 8, and 16. This value corresponds to how many times each X line will be scanned to determine the average. So how do we choose the best setting? This is a matter of taste, but will also depend on how fine your image measurements ultimately need to be. A noisier image will be much more complicated to segment and quantify later on. So ideally you want to generate an image that is as clean as possible. The OiVM core has some recommendations for this. Our recommendations depend on the master gain you set for each channel. For gain values less than 600 volts, no averaging is typically required. For voltages between 600 and 700 volts, we recommend a 2 to 4x average. From 700 to 800 volts, 4 to 8x. From 800 to 900 volts, 8 to 16x. And above 900 volts, 16x averaging. Since our voltage is currently set to 812 volts, let's choose the 8x averaging setting and scan a new image. You'll immediately see that this scan takes 8 times longer than the scan with no averaging. Remember, this is because each X line is scanned 8 times, and then the averaged result for each pixel is displayed. Once the scan is complete, let's put these images side by side and compare the result. The image on the right is our averaged image, while the image on the left has no averaging. This is a significant difference in quality, so depending on your master gain, it will be beneficial to use a reasonable averaging value. Alongside the averaging feature, ensure that your image bit depth is set to 16-bit. For whatever reason, Zeiss defaults this setting to 8-bit. If we expand the options, you'll see settings for 8-bit, 12-bit, and 16-bit. Ideally, you want to set this value to match the analog to digital conversion being done by the LSM hardware behind the scenes. Since the LSM 880 converts the analog signal from the PMT to digital in a 20-bit space, we want to set this to the highest setting available, 16-bit. This will ensure that our dynamic range is sampled as widely as possible, preserving the smallest variations in our detected signal. The final feature we want to discuss in the acquisition mode dialog is a useful feature for adjusting our final field of view, referred to here as zoom. While the objective magnification sets the general field of view, you can adjust this further in the software by a factor of between 0.5x and 40x with some changes to our scan area. Here, in addition to zoom, we can adjust our offset and rotation to optimize the final area we want to scan. The zoom is also important because it will affect the optimal frame size. Increasing the zoom by a factor of 2 will reduce the optimal pixel array size by that same factor. So for example, if we have an optimal array of 3440 by 3440, and we increase our zoom to 2, the optimal array will now be half that size at 1720 by 1720. You can use the zoom feature by manually entering the values or by using the interactive crop tool located in the Dimensions tab at the bottom of the image display. When we select crop, an interactive display pops up on the current image window. Here we can adjust the zoom, the rotation of our scan, or reposition the scan field. When we update the scan, the new area will be reflected in the image window. Click on Reset All to restore the default field of view. Now that we have optimized each of our detection channels and programmed in our desired resolution settings, we can begin acquiring our final images. 2D images can be captured via the snap button as previously discussed. 3D datasets can be acquired through the Z-Stack functionality. To collect a 3D dataset in Zen, activate Z-Stack from the multidimensional checkboxes below our primary scan buttons. Checking the box next to Z-Stack will activate the blue Z-Stack dialog in multidimensional acquisition. Expand this dialog to reveal its settings. You can even undock and reposition the dialog as you see fit. To collect a Z-Stack, first we need to mark the top and bottom focus positions of our sample. As a side note, if you have more than one fluorophore image, it is recommended to choose a representative channel to mark the stack positions. Using a single channel for this purpose permits the live view to update much faster, reducing exposure while the stack is being defined. Use the live function to begin updating the image. With the live scan running, position the focus away from the specimen by rotating the fine focus knob back towards you until the image within the scanned area goes just out of focus. Mark this position in the Z-Stack dialog by clicking Set First from the default First Last tab. While continuing to scan, Now rotate the fine focus knob in the opposite direction, focusing through the specimen until you begin to lose focus on the other side of the sample. When the image is out of focus, click Set Last in the Z-Stack dialog. Stop the live scan. Now our Z range is defined. From here we need to tell the software how many images we need to take within this range. Use the optimal function for this. Similar to the optimal function in frame size for XY resolution, the optimal function in the Z-Stack dialog sets the interval, or step size between the image, to precisely 1/2 of the optical section thickness for the longest wavelength fluorophore, reported in our channels dialog under pinhole. In this example, our optical section thickness at 1 error unit for this objective was 1.8 microns. So our Z interval would ideally be 0.9 microns. Click on the optimal interval. Now the Z-Stack dialog reports the range and total number of images required to sample that range accurately. To execute the stack, click Start Experiment just below our primary scan buttons adjacent to our multidimensional checkboxes. While our Z-Stack is running, we can change the viewing mode using the toolbar along the left-hand side of the image. From this toolbar, select Gallery to view all the slices together as they update. Once our stack is complete, we can save this dataset by navigating to the Images and Documents tab along the right-hand side of the screen. Here you will find a list of any unsaved documents, noted by the warning icon. Click the disk icon to give your file a name and save it to the hard drive. Please store all your images on the D or data drive on the host computer. Here you can create a folder for yourself or your lab. Name your file and save the file type as CZI. CZI is Zeiss's native image file format. This file format is essentially a TIFF file, however the header of the file contains all of the relevant metadata for how the image was acquired. This metadata will be important for your quantifications later on. If you choose any other format than CZI, this scan metadata will be lost. Once you have completed your imaging session, you must prepare the microscope for the next user, or power it down for the day. Position the microscope objective into the load position, and remove the sample holder from the stage. Store these holders in front of the computer monitor for the next user. Please check the iLab calendar to see if there are any users scheduled after you. If there are, you can leave everything running, including the Zen software, but please be sure to close any images you may have open. If you are the last user of the day, please power off the instrument and laser systems. To power off the LSM 880, exit the Zen software by navigating to File and selecting Exit, or clicking the X in the upper right-hand corner of the software. When you exit the software, Zen will prompt you to power off the lasers. Toggle the power state to off for every laser that is currently powered on. The argon laser will go through a 5-minute cooldown phase when toggled to the off state. You will hear the cooling fan for the laser running during this time. It will automatically shut off when the cooldown is complete, so please do not turn off the main power switch until this fan stops. Don't worry about the 405 laser, it will automatically shut off when the software exits. After the Zen program quits, give the computer about 30 seconds and then shut it down from the Windows Start menu. When the host computer is powered off, turn off the HXP 120, followed by the system's PC and component switches. When you hear the fan for the argon laser turn off, you can turn off the main power switch and the shutdown is complete. If you have any questions about this technology, or just want to discuss what options you might have for your own imaging experiments, please feel free to contact the OiVM Core through our website or via direct email. Thank you.